This is a revision video for the 6th topic in AQA GCSE Combined Science or Biology. These are pure factual recall questions based on the specification because recalling these key facts makes up 40% of the marks in GCSE Science. If you'd like to work through the questions before you watch the video, there's a link in the description below. Because this is a big unit, I've split it out into 2 videos. This one covers all the content for Combined Science, and then if you're taking GCSE Biology or what you might call Triple Science, you also need to watch the second video. Meiosis is a type of cell division. It's used to produce sex cells called gametes. And in humans, these gametes are haploid, which means they have half of the amount of DNA that's found in the rest of the body's cells. The gametes are different to one another and also to the parent cells. They share 50% of their DNA with the parent cells from which they were formed. There are 2 divisions involved in meiosis. This is different to mitosis and explains why it is that the cells that are produced only have half the normal amount of DNA. Gametes are sex cells. In animals, these are sperm and egg cells, and in plants, these are pollen and egg cells. Whereas sexual reproduction requires 2 parents who each produce gametes, asexual reproduction requires a single parent. The gametes produced by the 2 parents in sexual reproduction go on to undergo fertilisation, whereas in asexual reproduction, because there are no gametes, there is no fertilisation. This also means that in sexual reproduction, there's mixing of genetic information, whereas in asexual reproduction, there is no mixing, and the offspring are clones of the parents. Fertilisation is the process in which not just the 2 gametes fuse, but actually their 2 nuclei fuse. After this fertilisation, an embryo is formed, and the embryo will then go on to grow by mitosis, which is a different kind of cell division. DNA, or deoxyribonucleic acid, is a polymer. In fact, it's made of 2 polymer chains, which form a shape called a double helix. This double helix is made of 4 complementary nucleotides. Complementary means that they match up in pairs, rather like an enzyme and its substrate. The 4 nucleotides are called adenine, cytosine, guanine and thymine, although you can refer to them as A, C, T and G. Each nucleotide looks a bit like this. It's made up of a sugar, which is represented by the pentagon, a phosphate group and also a nitrogenous base. That's the A, C, T and G. In human cells, DNA is found in the form of 23 pairs of chromosomes. The genome is all of the genetic information in an entire organism. And the Human Genome Project had 3 key aims: to search for genes that were linked to particular diseases that were heritable, to understand and treat inherited disorders, and also to track historical migration patterns. A gene is a short length of DNA that provides the code for a sequence of amino acids which will go on to form the basis of one protein. An allele is a variant of a gene. This is a really common definition to get wrong, and so it's one that's worth practicing. Don't talk about flavours or anything else. The word you want is variant. If an allele is dominant, that means that it will be expressed, in other words, we'll be able to see it in the phenotype, even if there is just one copy of that allele. So in other words, we will be able to see it expressed even in a heterozygote. In contrast to this, a recessive allele will only be expressed when there are 2 copies present. In other words, it must be in a homozygote or we won't see it expressed in the phenotype. Homozygous means that there are 2 matching alleles of the same type. It's possible to be homozygous dominant or homozygous recessive. Heterozygous means that the 2 copies of an allele are not the same as one another. Genotype is the actual alleles that an organism has, whereas phenotype is what we see expressed. It's the observable characteristic. The majority of characteristics are coded for by more than one gene, but there are some examples of single gene characteristics. The ones that you should know about are fur colour in mice, red-green colour blindness in humans, cystic fibrosis and polydactyly. In the foundation tier papers, it's very likely that you will be given a pre-drawn Punnett square to fill in, but in the higher tier papers, it's quite usual for them to just ask you to draw one from scratch. So in this question, we're asked to draw a Punnett square for heterozygous parents, and we're looking at a disease that is recessive. So here I'm going to use A for my single letter for my gene, just because the capital letter and the lowercase letter are very easily distinguishable, so it will be clear to you which is which. So the top and the side of the Punnett square represent the gametes, so the sperm and the egg. And since my parents are heterozygous, each one of them will have 50% of the gametes carrying the dominant allele and 50% carrying the lowercase allele. So those are my gametes. And then in the main boxes of the Punnett square, I'm just going to match up. So here I'm going to have capital A, and here I'll have lowercase a. And then going left to right as well, here I'll have a capital A, and here I'll have a lowercase a. It doesn't matter if you've written your genotype with the capital letter second. The convention tends to be to always write the capital first, but you will get marks even if you've done it like this. So in this, we're then going to be asked what proportion of the offspring have the particular disease, or what probability it is. And because this is a recessive disease, I'm only going to see this expressed in the homozygote. So in other words, even if another offspring has one copy of the allele, they're not going to have the disease. So it's only going to be this individual out of the 4 that has the disease. So therefore my probability will be 1 in 4. Similarly, we could be asked to form a Punnett square for heterozygous parents who are carrying a dominant disease. And remember, this means that you only need one copy of the allele in order to have the disease. So again, my parents are heterozygous. Here I'm going to use the letter B because, again, it's very easy to distinguish which is capital and which is lowercase. And if I fill in all of the parts of my Punnett square, I have a very similar Punnett square because I've started with 2 heterozygous parents, but crucially this disease is dominant, and so that means that I only need one copy of the allele in order to have the disease. So obviously my homozygous dominant individual will have the disease, but also so will my 2 heterozygous individuals. So 3 out of 4 have the disease. Cystic fibrosis is a disease of the membranes, and it causes them to make a lot of sticky mucus, and this can cause problems particularly with the respiratory system but also with the digestive system. Cystic fibrosis is caused by a recessive allele, so you need 2 copies of that cystic fibrosis allele in order to have the cystic fibrosis disease. Polydactyly is another genetic disease in which individuals have additional fingers or toes, and it's caused by a dominant allele. When it comes up, embryo screening often forms the basis of an extended question where you're asked to evaluate both sides of the story and reach a conclusion. But these are meant to be purely factual recall questions, so we're just going to focus on what some of the advantages and disadvantages might be. The major advantage of embryo screening is that you're reducing the possibility of suffering. So you do embryo screening when there's a particular genetic disease that runs in the family, and what you're trying to avoid is having a child born who is going to have perhaps a very short life, perhaps a very poor quality of life, because they're born with a disease that is really going to restrict them and limit their life. The second thing is that you're also going to reduce the costs associated with that disease. So often, even if these children are only alive for a short amount of time, they're going to need round-the-clock care, they may need further surgeries or expensive medical treatments, and trying to keep them alive is going to be very financially expensive. Also, when you do embryo screening, you don't just screen a single embryo. Often the parents will produce a large number of embryos, and then these will all be screened and 1 or 2 will be selected to be implanted into the mother. That means that there are spare embryos, some of which may be entirely viable and some of which may have the disease that we're trying to avoid. And so those are available for scientific research or possibly for stem cell treatments. In terms of disadvantages, there are a wide number of ethical issues, and it's important if you're stating these that you specify what they are, because sometimes just saying ethical issues won't be enough to get you the mark. As we've said, there's often a large number of embryos generated by the parents before screening, and the rejected embryos, if they're not used for scientific research or stem cell treatments, may simply be destroyed, and some people have a moral objection to that. There's also a concern that this same technology that is currently used to screen for really debilitating genetic diseases could be used to screen for something as simple as eye colour or sex, and people are uncomfortable with that idea of designer babies. There's a question about whether screening and trying to avoid someone being born with a particular disease causes discrimination and prejudice towards those who are already living with that disease. And also there's the fact that the embryos are unable to consent to this medical procedure because they're not even fully fledged humans yet. Additionally, you can also talk about the fact that embryo screening can be very expensive, and also it increases the risk of miscarriage. There's also the possibility of a false positive. This is where the scientists believe that an embryo has the genetic disease even though they actually don't. So there's a possibility that that embryo is destroyed when it could have been a viable human being who was perfectly healthy. With the exception of the gametes, cells in humans contain 23 pairs of chromosomes. But only one of those pairs of chromosomes is concerned with sex determination. In the vast majority of biologically male humans, the genotype is XY, whereas in female humans it will be XX. Variation describes differences in characteristics of individuals within a population. It may be caused by differences in genetics, the environment, or a combination of both the inherited genes and the environment. Blood type is caused by genetics, whereas a scar or a skin tan are caused by changes in the environment. It's taken over 3 billion years for all of the life present on Earth today to evolve from simple life forms. Evidence for natural selection comes both from the fossil record and from the evolution of antibiotic-resistant bacteria. Bacteria are able to evolve so rapidly because they have a very short generation time, and also because they're haploid. Because they don't have a second copy of every gene, this means that any changes to their genetic material will result in an identifiable phenotype that selection can act upon. New strains of bacteria arise as a result of mutation. One example of antibiotic-resistant bacteria are MRSA. Bacteria such as these may become widespread when, firstly, there's mutation. This leads to variation in the population. In other words, there are some bacteria that are better at resisting an antibiotic than the general population. There's then a selection pressure. In this instance, somebody starts taking the antibiotic. There's a selection advantage. Some bacteria survive, whereas others do not. But crucially, not only do they survive, but they survive and breed or reproduce. When they do this, they're able to pass on their alleles to the next generation. And so the number of resistant bacteria carrying those alleles increases. Eventually, due to a lack of immunity and effective treatment, those bacteria are able to spread throughout the community. To minimise populations of antibiotic-resistant bacteria, firstly, it's important that if you're prescribed a course of antibiotics, that you take the full course. In other words, if you're prescribed 7 days, you don't just stop taking them after 3 because you're feeling better. The second thing is something that doctors can do. They should not prescribe antibiotics for viral infections or even for particularly mild infections that you should be able to get over on your own. Finally, it's important that the antibiotics that are used in agriculture, in other words, things that we feed to our chickens or to our cows, are not the same antibiotics as the ones that are being prescribed to humans. It can often be challenging to produce new antibiotics because developing them takes a really long time, and it also takes a lot of money. Fossils are the remains of organisms which over the course of millions of years have become preserved in rocks. There are a number of different ways in which they can be formed. Firstly, it may be that certain parts of the organism have not decayed. This is often the hard parts like bones and teeth, and it may be that there's been a lack of one of the conditions that's necessary for decay. For instance, there might not have been enough oxygen, or the temperature might have been too low. These parts are often then preserved when sediment lies on top of the hard parts that haven't decayed, and then over millions of years it hardens into rock. Alternatively, it's possible that some of the parts, such as bone, may be gradually replaced over time with minerals in a process called mineralization. Sometimes, rather than the organism's body being preserved, it will be other traces of them, such as their footprints or their burrow or rootlet traces. The early life forms that evolved on Earth were soft-bodied, and therefore they tended to decay very easily because there weren't bones and things to be left behind that would take a longer period of time to decay, and therefore they're just not found in the fossil record. Extinction happens when all of the individuals of a particular species have all died out, so there's no possibility of that species ever coming back. Natural selection is quite a complicated process with 5 key steps that you need to remember. Firstly, mutation occurs, and this causes changes to the DNA. This means that within a population, individuals will show variation for a particular characteristic. They won't all be exactly the same as one another. The individuals that have the characteristics that are best suited to the environment they're living in are the most likely to survive. And crucially, when they survive, they're most likely to reproduce. This means that they can then pass on their alleles to the next generation. If this reproduction step doesn't happen, then it doesn't matter that they survive for longer. The next generation are then going to have more of this characteristic because the number of individuals that have these alleles will be greater. Speciation occurs when 2 populations of the same species become so different to one another that they're no longer able to interbreed to have fertile offspring. In other words, even if they can breed and produce a baby, that baby is not able to have babies of its own and produce another generation. The steps for speciation to occur start off with variation. If there's variation in the population, then there's something for evolution to act upon. The second step is isolation. In other words, the 2 different populations aren't able to interact with each other. This might happen if they're split up onto different islands, or maybe if a new river forms that splits the population in 2, or maybe just because they've been migrating away from one another and the distance is such that there's no longer any interbreeding. Then, because they're in different places, there are different selection pressures acting on them. So that might mean that maybe they've ended up on different islands and there are different food sources on the 2 islands, or it might mean that the climate is different. Because there are those different selection pressures, different individuals will have an advantage, and so we get what's called divergent evolution. This might mean that whereas on one island the birds are growing a really big beak for breaking apart hard seeds, on another island the birds are developing a very skinny beak that will allow them to catch insects that are in rock crevices. Finally, the 2 populations become so different to one another that they are no longer able to interbreed successfully to produce fertile offspring. Selective breeding is the process where farmers or breeders or scientists develop new varieties or new breeds by controlling breeding. Basically what they're doing is looking for desirable traits, in other words, things that are going to make the organism that they're breeding more popular or more valuable, and they're only allowing the individuals that have those traits to breed. To start with, the farmer or gardener or scientist selects a male and a female that have the most desirable traits. So that might be a cow that produces the most milk and a bull from a mother who also produced a lot of milk, or it could be picking 2 plants that have really attractive flowers, or whatever it is. So they're going to take a male and a female that have those traits in their genetic lines, and then they're going to breed them together. From the offspring, they're going to look and identify which of the offspring also have this really desirable trait. And then they're going to repeat the process over a series of generations. There are loads of examples that you could give for where selective breeding is commonly used, but the 4 from the specification are disease-resistant crops, animals that produce more meat or produce more milk, friendly domestic pets, and also plants that produce large or unusual flowers. The major disadvantage of selective breeding is that, particularly if we do it in a way that's as simplistic as we've described in question 54, you're going to end up inbreeding. This basically means that individuals that are too genetically similar to one another have been bred together, and therefore there's going to be an unusually high prevalence of genetic diseases. In reality, when we undergo a breeding programme, it's made slightly more complicated than we've described, and family trees attract really, really closely to prevent inbreeding from happening. In transgenic genetic engineering, we identify a desirable trait in one organism and isolate the gene that's responsible for that trait, and we then transfer it to a second organism, effectively editing its genome to mean that that second organism can also express the desired trait. There are loads of different examples of genetic engineering, but the 2 named in this part of the specification are introducing disease resistance to crops and also increasing the yield by increasing the size of fruits. In modern medicine, we produce a huge amount of insulin for people who have type 1 and type 2 diabetes by using bacteria that are able to express the human insulin gene. If you're sitting the higher tier, then you need to be able to describe how this genetic engineering is carried out. First, the gene for the particular trait of interest is isolated, and this is done using special enzymes called restriction enzymes. Then the gene is transferred into the organism of interest, and this is done using something called a vector. This could be a plasmid if we're using bacteria, or it could also be a viral vector. This is done at a very early stage of development, and the reason for this is that it ensures that all of the cells of the target organism contain the modified gene, rather than just some of them, which is what would happen if we did the transfer at a later stage of development. People do have some concerns about genetically modified crops, specifically the impact on other organisms living in the same environment, so that could be plants that need to compete with a particular crop or insects that might eat it. And also some people are concerned that because this is relatively new technology, there haven't been a huge number of long-term studies about the impact on human health of eating these genetically modified organisms. The traditional 5-kingdom classification system was developed by Carl Linnaeus, and this includes 7 ranks: kingdom, phylum, class, order, family, genus and species, and it's the latter 2 of these that together make up the binomial name of a species. The classification system has now been overhauled, and this was as a result of 2 key pieces of evidence. Firstly, improved microscopy means that we've been able to look at the really fine detail structures within cells. Secondly, we also now know far more about biochemical processes. The modern classification system splits all organisms into 3 domains: the eukaryotes, which contain a true nucleus, the prokaryotes, which are sometimes referred to as the true bacteria, and also archaea, which were initially thought to be primitive bacteria, although we now know that they are as different from the true bacteria as the true bacteria are from eukaryotes. Evolutionary trees can be used to show the evolutionary relationships between different organisms. In other words, how closely related to one another are they? Thank you very much for watching, and I hope that you found this useful in your revision. If you're taking GCSE Biology, then don't forget that there's a separate follow-up video with all of the triple science content. If you did find this useful, then don't forget to like and subscribe for more GCSE Biology content coming soon.